In 2002, a woman in Washington state was applying to receive welfare benefits for her children. A routine DNA test was required to confirm she was the biological mother. The test came back negative. According to her DNA, she was not the mother of her own children. She knew she had given birth to them. There were witnesses, medical records. But her DNA said otherwise. Investigators initially suspected fraud. They were wrong. What they eventually discovered was something that medical science had barely begun to understand. She had two completely different sets of DNA in her body, one in her blood, a different one in her reproductive organs. The woman's name was Karen Keegan, and her case changed what doctors understood about human genetics. She was what is called a human chimera. From the Greek mythological creature, part lion, part goat, part serpent. In biology, a chimera is an organism containing two or more genetically distinct cell populations, both derived from different original cells, meaning different DNA, in the same body. This is not a mutation. It is not a disease. It is a condition where two genetically distinct individuals essentially merged into one, usually during very early embryonic development. The most common cause? Twin embryos that fuse in the very earliest stages of development. One of the twins essentially absorbs the other at such an early stage that no separate twin ever develops. The result is a single individual who carries the DNA of both, distributed differently in different tissues throughout the body. Some tissues may be predominantly one genetic profile, others predominantly the other. In Karen Keegan's case, her blood carried one genetic profile. Her thyroid, hair follicles, and skin cells showed a different profile. Her reproductive organs, the source of eggs that became her children, carried the second profile, which is why her children didn't match her blood DNA. They matched her other DNA. A DNA she wasn't looking for because she didn't know she had it. The diagnosis took months and required testing multiple tissue types across her body. It was one of the first documented cases of human chimerism with such dramatic legal implications. When Karen's case was published, many researchers assumed this was extraordinarily rare, a once-in-a-generation medical curiosity. They were probably wrong. The more researchers have looked for chimerism, the more they've found it. In bone marrow transplant patients, chimerism is actually the goal. The transplanted marrow creates a second blood-producing genetic population alongside the original. Intentional medical chimerism. Thousands of patients are living chimeras today, but natural chimerism, the kind Karen had, turns out to be surprisingly common too. Studies of vanishing twin syndrome have revealed a key insight. Vanishing twin syndrome occurs in an estimated 20 to 30% of multiple pregnancies. One embryo fails to develop and is reabsorbed. But sometimes cells from the absorbed twin persist. In the surviving twin's body, genetically distinct, integrated into different tissues because the rates of twin pregnancies, including vanishing twin pregnancies, are higher than most people realize. The actual rate of natural human chimerism may be significant. Some researchers estimate that a measurable percentage of the population may carry cells from a twin they never knew existed. Most of them will never know, because standard medical DNA testing only tests one tissue type, usually blood. If your chimeric cells are in your liver, or your skin, or your reproductive organs, a blood test won't find them. You would simply walk through life with two complete genetic profiles and never find out. The implications of chimerism extend well beyond medicine. DNA evidence is the gold standard of modern criminal investigation. It is treated as virtually infallible in courts worldwide. A DNA matched to a crime scene sample is considered extraordinarily powerful evidence. Chimerism breaks this assumption, A chimeric individual's DNA at a crime scene depends entirely on which tissue left the trace. Blood at the scene might match one profile, skin cells a different profile. A saliva swab from the suspect might match neither. A known chimeric person could potentially commit a crime, leave DNA evidence, and have a DNA test come back negative, not because the test failed, but because the test was technically accurate. It tested a different genetic profile from the one that left the evidence. Legal systems built on the assumption that each person has exactly one DNA profile are unprepared for this. Most legal systems have not formally addressed chimerism as a challenge to DNA evidence admissibility. It remains a gap between what biology can do and what the law assumes biology does. There have been real cases where chimerism complicated criminal investigations. In 2004, a chimerism case in the U.S. arose during a paternity dispute. The man's blood DNA didn't match the child, but his sperm DNA did. He was almost denied legal paternity of his own biological child because of his chimerism. These cases are currently rare enough that courts can handle them case by case. But as DNA testing becomes more widespread, more chimeric cases will emerge, and the legal system will need to formally grapple with a biological reality it has mostly been able to ignore. People walking around with two complete sets of DNA and not knowing it, sometimes discovering it in the most unexpected ways. A woman told by DNA evidence she wasn't the mother of her own children. A man almost denied paternity of his biological child, and the possibility that you might be one. If you want more of what the human body is actually doing, subscribe. Next week, a nuclear test so powerful it created a second sunrise visible across 3 continents.